In this video, I want to describe the graded membrane potential changes that occur in neurons in response to input, which we just call graded potentials for short. So I've drawn a blown up neuron here. We have a soma in red and an axon in green and two dendrites in blue. And recall that resting neurons, that is neurons that aren't receiving any input, usually have a stable charge separation across the entire membrane, where there are a layer of positively charged ions, also called cations, on the outside of the neuron membrane, and a layer of negatively charged ions, also called anions, on the inside of the membrane. And then we call the outside 0 just to set it as a reference, and that the resting membrane potential of neurons may vary, but it's often around negative 60 millivolts. So let's show that on a graph here. Let's say we're looking at this piece of membrane, and on the x-axis we'll put time, and on the y-axis, we'll put the membrane potential in millivolts. And so let me put right in the middle here this negative 60 millivolts. It's a common neuron resting potential. And that when the neuron is at rest without inputs, most neurons just have a stable potential at their resting membrane potential, where it's not changing over time without input. Now, inputs from certain types of stimuli may increase or decrease the membrane potential of the neuron a small amount for a brief time before it returns back to the resting potential. These transient membrane potential changes are called graded potentials, and they tend to occur in the dendrites of the neuron and in the soma of the neuron. And the size and the duration of the graded potentials is determined by the size and the duration of inputs, both excitatory inputs and inhibitory inputs. Graded potentials do not pass into the axons of most types of neurons. Instead, most axons have a different membrane potential change called an action potential. Action potentials start at the area called the trigger zone, which is the initial segment or the start of the axon. And they start when the combined effect of the graded potentials at any moment in time brings the membrane of the trigger zone across a certain value called the threshold potential. So let me just draw that with a little dashed line here. And this threshold potential will vary between neurons, but somewhere around negative 50 millivolts would be a common threshold potential. So that if the membrane potential at the trigger zone can be moved from the resting potential, which is often around negative 60 millivolts, over the threshold potential, which is often around negative 50 millivolts, then a totally different potential change will happen called the action potential that will shoot all the way down the axon. Now, this adding together of graded potentials is called summation, and summation at the trigger zone is how neurons process information from their inputs. Most neurons respond to inputs from other neurons in the form of neurotransmitter molecules that are released at synapses. So that if this is the axon terminal of another neuron, it may release neurotransmitter at the synapse where these two neurons come together, which will bind to little receptors on the membrane of this neuron, in this case here on a dendrite, and this will produce some kind of graded potential. Now, we'll get into the details of this more in other videos, but this is the most common type of input that a neuron will receive. And depending on the neurotransmitter and depending on the receptor, this may be an excitatory input or maybe an inhibitory input. Now, some other types of neurons and neuron-like cells that are sensory receptors may also generate graded potentials from physical stimuli, such as light or odorant molecules. Graded potentials produced from a synapse are called synaptic or postsynaptic potentials. And those generated by stimuli and sensory receptors are also called receptor potentials. A graded potential like this one that moves the membrane potential to a less negative number or closer to 0 is called a depolarization. Depolarization. Because now the membrane is less polarized, it has less charge separation. These are also called excitatory potentials because they move the membrane potential closer to the threshold, so they increase the likelihood that an action potential will be started at the trigger zone. A graded potential like this one that moves the membrane potential to a more negative number, farther away from 0, is called a hyperpolarization. Hyperpolarization. Because it's increasing the polarization or the charge separation of the membrane. Hyperpolarizations are also called inhibitory potentials because by moving the membrane potential farther from the threshold, they're decreasing the likelihood that an action potential will be started at the trigger zone. Two important properties of graded potentials are that they decay with both time and distance, so that their effect is brief and local. Graded potentials decay with time, just like I've drawn here. The membrane potential changes for a brief time and then it returns to the resting potential, unless there's more input. And because graded potentials decay with time, if two graded potentials happen that are separated by enough time, they won't have any effect on each other. For example, let's say that this depolarization happens and is finished before a second depolarization over here occurs. Since this one was already done, already fully decayed, these two had no effect on each other. But if two depolarizations happened right around the same time, their effects can add together. They have additive effects. And you can get a depolarization twice the size. We call this process temporal summation, or adding together of graded potentials in time. Graded potentials also decay with distance as well as with time. So let's look at this depolarization, and let me just move it over here. Let's say that this synaptic potential or postsynaptic potential is a depolarization. Let me say right at this piece of membrane, we get about this size of a depolarization. As the depolarization spreads across the membrane, it's going to decay in size. So let's say maybe we check in with it here, and at this piece of the membrane, now it's a smaller size than it was when it started over here. And as it continues spreading across the membrane, maybe if we check in with it over here, it's now actually quite small. So that by the time it gets to the trigger zone, where the decisions are made to fire an action potential or not, the depolarization that started way over here may not have much of an effect on the membrane at the trigger zone. Similar to the concept of temporal summation is the concept of spatial summation, that if two graded potentials happen far enough away from each other, they may have no effect on each other. For example, let's say that there's another excitatory input way down here at this dendrite that causes a depolarization. Just like this depolarization, as this spreads across the membrane, it's going to decay so that it'll get smaller with distance. So that maybe by the time these two reach the trigger zone, they decayed entirely so that they have no effect on each other. But if instead you had two kinds of excitatory input very close to each other on the membrane, then those two depolarizations could have spatial summation. They could add together in space so that you could get a depolarization twice the size. The same would be true for hyperpolarizations. You can have temporal and spatial summation of hyperpolarizations. You get hyperpolarizations that are larger in size. So what would happen if you had an excitatory input and an inhibitory input at the same time and place? Well, instead of getting both a depolarization and a hyperpolarization, what you may get is no change to the membrane potential. They may cancel each other out and leave the membrane potential at the resting potential. Now, one effect of the fact that graded membrane potential changes decay with distance is that the closer an input is to the trigger zone, the greater effect it will have on the likelihood of an action potential being fired down the axon. Because if a graded potential starts closer to the trigger zone, it will decay less by the time it gets there than a graded potential that starts farther away and decays more with greater distance. Therefore, a synapse that's closer to the trigger zone will have a greater influence on the behavior of the neuron in terms of action potentials being fired than a synapse that's farther away. For example, here, way out at the end of a dendrite. One last thing that I want to mention is that synaptic potentials like these tend to be quite small in size. And in fact, I've drawn these too large because they're usually less than 1 millivolt in size. Therefore, most neurons require the temporal and spatial summation of many synaptic potentials to move the 10 millivolts or so that usually separate a typical resting and a typical threshold potential for any particular neuron. So that as all the different synapses that are connecting this neuron to lots of other neurons in its network are creating all these synaptic potentials, the membrane potential of the dendrites and the soma is constantly moving around and wiggling around off the resting potential until there's enough excitatory potentials, enough of these depolarizations that are being summed in space and time to cause an action potential to be fired down the axon. So some very complex processing of information from all these inputs can occur because of these graded potentials.